My name is Mei Wong, and I will be teaching you about the endomembrane system and endomembrane trafficking. So this is our cell, and the parts of our cell contain the plasma membrane and the Golgi apparatus. Then here we have our endoplasmic reticulum. Here's the rough ER and the smooth ER. Then the cell contains a lysosome, which breaks down and recycles waste within the cell. And then the cell has a mitochondrion, which is the powerhouse of the cell. So endomembrane trafficking is the flow of products between different organelles through a cell. And endomembrane trafficking starts here in the nucleus. The nucleus is the control center of the cell, which contains the cell's DNA, which is a double helix composed of nucleic acids. So you have adenine, thymine, cytosine, and guanine. And the adenine pairs with the thymine, and the cytosine pairs with the guanine. Okay. Then the start of endomembrane trafficking begins with DNA replication, where DNA polymerase unwinds a portion of the DNA, creating a template strand, which is this strand, which is composed of codons. Codons are a triplet of the nucleic acids. So there. And then this will then lead to transcription, where the DNA template, which is this top one right here. So the DNA template is copied into mRNA, which is this strand in the bottom, with the exception of uracil, which is right there. In mRNA, instead of having adenine pair with thymine, it pairs with uracil, but the template strand still has thymine. So that happens in the nucleus. And then from there, the mRNA moves out of the nucleus to the ER. And in the cytoplasm of the cell, the mRNA is read by ribosomes and tRNA to form amino acids. So this is translation. So these are the tRNA, and then these are the amino acids that they produce. And the tRNA contains an anticodon that pair with the codons in the mRNA strand, and the amino acids formed create a polypeptide chain. So that happens in the cytoplasm. And then as that travels through the ER, so as the polypeptide chains of the amino acids form until there's a stop codon reached, and before it goes through the smooth ER, the polypeptide chain goes through post-translational modification, which is the process where the polypeptide chains are cut or folded to form a protein. So that would be protein synthesis. The ribosomes are responsible for protein synthesis. And then from there, the proteins that are formed move through vesicles to the Golgi apparatus, and the proteins go in through the cis phase, which is this part of the Golgi apparatus. And then as they go through the Golgi apparatus, this part of the Golgi apparatus is called the cisternae. So right there, it's just a bunch of folded vesicles. And then as the protein goes through, it goes through the cisternae, it will come out of the transphase, which is this side of the Golgi apparatus, and they will exit through secreting vesicles in the plasma membrane. And then once they reach the plasma membrane, they will exit the cell, and this is a process called exocytosis. And that's endomembrane trafficking.